think about it today, Friday we'll try to have a decision, right? Before we come back. So we're putting the electrocardiography aside for a couple of 3 or 4 lectures, and we're going to start talking about cardiovascular disease. We're starting with uncommon diseases that are genetic in origin. So not a lot of people have these, but if it runs in your family, boy howdy. So by the end of next week, we'll finish. We're going to focus in here on hypertrophic cardiomyopathy because it makes several teaching points as an example. But by the end of next week, we're going to move on to common. 3/4 of all cardiovascular disease is atherosclerosis. So we'll move on to the really common cardiovascular diseases and what drive them. And these almost all come not from single genetic loci, but polygenic dispositions interacting with a bad environment that turns them on. All right, does that make sense? So here's today's lecture. We're going to talk a little bit, give you some examples, 2 or 3. There's hundreds of these, and I don't want to teach you about a disease that occurs twice in a million. It's not useful unless you have it. So I'll mention the basic patterns of these genetic cardiovascular disease, and then we're going to focus, come down on one of them, hypertrophic cardiomyopathy. This is the disease of overgrowth of the heart that predisposes young athletes. When you see a young athlete drop dead, there's a better than 50% chance that it was this. And as a segue between these two, I need to review for you what the normal growth of an athlete's heart should look like with training. So then we're going to start working on how do we distinguish these 2 different kinds of enlarged hearts. So, on to genetically predisposed hearts. So, when a genetic variation is the prime determinant of serious cardiovascular disease, by the way, because it's inborn, you often see it young in life, right? You don't have to wait. Like atherosclerosis, it takes 40 years to destroy the arteries you were given, but this stuff can happen fast. One of the great problems with these diseases is because they develop asymptomatically, very often the first indication is something catastrophic like a heart attack or death. Some examples are dominant inheritances, familial hypercholesteremia, and I'll show you an example of that. There's also, I can say SNPs to you, can't I? Single nucleotide polymorphism. So if there's a critical protein that was inherited in mutation that impairs its normal function, then you can get strange results from that. Channelopathies and cardiomyopathies are due to SNPs. So here's the 3 inherited ones we're going to talk about. Diseases or conditions, genetic conditions that accelerate atherosclerosis. It's a common disease, but it can be greatly accelerated if you have the wrong genes. Channelopathies tend to precipitate arrhythmias. That's where a single protein in a channel is defective, and so you wind up with a prolonged refractory period. That would do it. And then finally, we'll get to cardiomyopathy, hypertrophy, which I want to talk about. So here's a nice Texas mom. How old is she? 40 years old. She's a rancher and a photographer, and that's her boy. No one, when she went to the ER with chest pain, sweating profusely, no one could believe she's physically active, goes to aerobics. How can a young woman like you with estrogen in your system be having a heart attack? But she was. Notice the headline, saves her son's life. She had a heart attack. How could that save her son's life? Well, because they measured in her, say she was a female, that's a female, with an inherited, it was filled in, it means you inherited cholesteremia. And so her child turned out to have, it's an 8-year-old or 12-year-old, cholesterol above 400. Well, we know what happens with that. Around 30, you'd have a heart attack. And so if you look through this, so you guys are familiar with cladograms, familial inheritances, so grandma, grandpa, first generation, second generation. Notice granddad was the carrier. He had his heart attack at age 47. That's what this circle means. Then you look through here. By the way, it applies, looking at this, you see the males get it It's not an excellent characteristic. Females can get it just fine. It's just that's how it happened in assortment for this family. Notice those who get it, have very high cholesterol. So that's what the third line is, cholesterol. And they tend to have heart attacks. They had a heart attack at 45, heart attack at 38, and it killed them. Removed from the gene pool. Notice then that we need a male and didn't. So let's go down to the children, the grandchildren. There's a male without it, and his total cholesterol is 94. Total cholesterol 130. Total cholesterol, see, that's fine. Remember, these are children, if you look here, these are young, so that's the age. Now, if you did that, here's a 13-year-old with cholesterol 344. That's why we have the National Cholesterol Education Program, government-funded program, to try to get every person in the United States to have cholesterol measurement before the age of 30. Because you can identify this and take action, because we know what's going to happen if you keep that alone. It'll be just like ball. Does that make sense? So that's an interaction where a single genetic dominant characteristic is driving you towards a disease. Notice that's one of the 5 primary risk factors for atherosclerosis is a high LDL. This gene gives it to you. If I remember right what the gene is, I can't quite remember, but I'm pretty sure it's a genetic disablement of the LDL receptor on the liver. So the liver is actually making normal amounts of LDL, but it can't take it out of the circulation and reprocess it like it ought to. I'm pretty sure that's what that is. And do you have any nutrition majors? I forget. Is anyone? Cool. Okay, so this is your disease. I mean, not personally, but professionally. Remember how the sulfur-containing amino acids, that they're broken down. So methionine and cysteine, they are processed like anything else, and waste is dropped into the urine. Well, there's an intermediate called homocysteine. And homocysteine turns sort of like acetaldehyde when you digest alcohol. Acetaldehyde is very toxic. It has a lot of side effects. That's what hurts people with excessive ingestion of alcohol. Well, homocysteine turns out to damage the arterial lining, the endothelial lining. So if you've inherited a SNP in one of the enzymes that breaks down methionine or cysteine, you could wind up with excess homocysteine in your blood. These people have their heart attacks in their teens and 20s. If you're, this is sorts, heterozygotes have a halfway, I forget the kind of inheritance this is. Homozygotes who both have both alleles as the damaged one, they'll have their heart attacks in their teens and 20s. Very hard to recognize someone having a heart attack who's a young, healthy woman or man. It's really hard for the doctor to think, could this be homocysteine? And then the heterozygotes are predisposed to have heart attacks earlier in life. Makes sense? Again, these are not common, but so a final example of an inherited disease are, genetic diseases in general. So when athletes drop dead, there's really 2 choices. The one is self-medication with sympathomimetic drugs. So if you're amphetamine or something to get up for the game, that actually is a significant subfraction of deaths on the athletic field. But the majority of sudden deaths in athletes are inherited diseases. So here's the example of an 18-year-old captain of a basketball team who's just shooting hoops in PE and he drops dead. That ain't right. That's not how it, many of you have shot hoops in PE and you're still alive. Something was wrong. It was an inherited cardiomyopathy, which is where we're heading. So here's there's a laundry list of inherited or cardiovascular, genetic cardiovascular diseases, almost in order of common. This is the most common, we'll see in a moment, so we're going to focus on it. Notice that arrhythmia, that kills you with arrhythmias. Both of these are good examples of channelopathies, where you inherit a SNP in one of your membrane proteins, and as a result, you will repolarization. You're in that partial repolarization sensitive period for a longer period of time. And so the likelihood that you're going to trigger a circus arrhythmia in your ventricle goes way up. I told you we lost a young man in the dorms who took a friend's Adderall to try to study for an exam, and he had long QT syndrome. And that's tragic. It's just tragic. It should not happen because it's so obvious. Someone did not educate him very well about what his disease was. By the way, Adderall is amphetamine, and it does not help with short-term learning. It makes you agitated and dance in the bottom. Don't take your friend's Adderall. You guys understand ADHD, when they take it, if you have a brain that's ADHD, you take amphetamine and they calm down and focus. And in fact, that's called a paradoxical response to amphetamine. And it's what defines, it's what confirms the diagnosis of ADHD. You can see how the layperson can say, well, you take that, it makes you calm and focused, I want it. Well, all it's going to do to you is make you sweat and dance on the wall. Sorry, a little commercial. So here's a visual of the last slide again. So here's cardiomyopathies. These are all the sudden deaths in athletes. And notice that if you figure that most congenital abnormalities are genetic, the cardiomyopathies are all genetic, and at least 10 out of 14% of these arrhythmias are genetic. It's about 50 to 60% can be viewed as inherited for these sudden deaths. All right. Oh, nice. Any questions about that? So there are, you don't have this. Your electrocardiograms and your histories would tell me more about it, but it's just vanished. They're rare diseases. However, if they run in your family, then it really matters. I have a few friends who have inherited hypercholesteremia, and that means you have to live differently quite early. By the time they're 25, they're taking cholesterol-lowering medications, but they can have a full life with no heart attacks. You just got to work at it. Does this all make sense so far? So, on our way to looking at cardiomyopathies in more detail, let's just review quickly, because training is supposed to make big hearts. Leo, this is your question about big hearts. How do we tell the good from the bad? And so you've seen this slide before. I'm just going back to, these are echocardiograms. The best way to measure the wall thickness or the valve function is an echocardiogram. We see that in the trained person, there's a bigger end-diastolic volume, so there's more filling. And then upon contraction, you can visibly see a thicker wall. So the wall thickens in proportion to the increase in the stretch on the heart and end diastole. That's the normal. There's 4 attributes. It is adaptive. So this actually makes you function better, not worse. It is symmetric, and that's 2 kinds of symmetry. First off, the hole and the wall grow in proportion, so that you wind up with a nice balance. We should really look at it and systole. You should always have at least 2 and a little bit more wall thicknesses in terms of how big is the hole at end systole. So it's symmetric. The other symmetry is that notice how this is bigger all the way around. That's important too. It would be strange, wouldn't it, if just one side of the heart had a big plum-shaped. Yeah, so 2 kinds of symmetry. Hole to wall, and then the wall is evenly hypertrophied. Reversible means if the athlete stops training, this goes away. We did a study on the Sacramento Kings with Dr. Longhurst in the cardiology department, where we followed them in and out of the season for like 3 years. You could literally watch their hearts grow and shrink. The ones who maintained high level of training off season, it was a smaller phenomenon. But the ones who got in shape and out of shape, they really changed the dimensions of their heart through time. So reversible. And then finally, normal hypertrophy doesn't mess up, to use a technical term, repolarization functions. The heart relaxes quickly, coronary flow is not obstructed, repolarization happens as much faster as the depolarization happens, so they stay in cadence. So all the diastolic function is preserved in a normal hypertrophy. So it turns out the way to tell an abnormal hypertrophy is if it fails any of these. That's really the best way. Yes, opinion?
Could you talk about the adaptive?
Again, well, adaptive would mean that as this this change grows, the person actually functions better. Their cardiovascular system, they have a higher VO2 max, they can do more work. You'll see with cardiomyopathy, the bigger the heart grows, the worse they function. They start passing out and falling on the ground. And so the more, here, active would be, it enhances your function, the growth. You'll see with disease, when the growth gets big, it actually makes you worse and worse and worse. Good question. So let's just really briefly remind ourselves what's happening in the normal heart as it grows. So here are two myocytes, cardiac myocytes. You really can't see that. That might be the nucleus and that might be the nucleus, but I'm kind of making that up. Notice you can see the strings of the myofibrils inside. Notice they're relatively short and they're single nucleated compared to the skeletal muscle. And I really like this next one. Oops, I ain't right. So watch this little video. Go to the myocyte, go. If you have a clock, you can time it, and it comes out that in a 6-second period, it's contracting twice. That comes out to a heart rate of 20. That's exactly what a myocyte, right? You just put them in a plane, they'll do that. I think that's cool. Remember that then, when you do training, each individual myocyte, remember you don't have more cells, but each myocyte grows bigger in dimension as it adds more myofibrils. So each one of these becomes bigger. Another thing that skeletal muscle can't do is they will actually add sarcomeres end on end. If you think about it, if this dimension, this end-diastolic dimension is getting bigger, you don't get more cells. So how are you getting all the way around this larger circumference? You never thought about this, but if you think about it, it means that each cell is actually becoming somewhat longer. And this is unique to the heart. Skeletal muscle doesn't do this because the bony distances don't change once we're adults. Remember I told you that. Anyone remember the functional property that a longer, if you had sarcomeres end on end, the property that would grant?
Faster.
Faster. So how cool is that? That exercise training, just like exercise training in a muscle, we know it's going to increase power if it's a thicker myocyte. So here's a trained cardiac muscle. I can't remember if it's a mouse or a rat, but there's the trained cardiac muscle will be stronger. But this is what's cool, is the Vmax actually goes further out. Oh, velocity, here's peak velocity, did it graphic first. Here's max velocity, there's the Vmax, and so Vmax is actually increased. Sir?
Do new blood vessels accompany the new?
You know, that's a really good question, and I'm going to give a general answer since I haven't seen the actual study. Capillary density is so maximized in the heart to begin with, it's 4 or 5 times what it is in the skeletal muscle. And so it's almost like it's installed at the high end and the heart catches up to it in muscle growth. Kind of like the lava.
Is the cardiomyocyte unable to add sarcomeres parallelly?
Oh no, it's adding both. That's what's so cool, is if you go back to this one, Mary, so this thing under training gets this way, side by side growth in the myofibrils, and it also has a length growth. So it's very cool. It's altogether a supercell. So human hearts are supposed to differ in size. So if we're going to talk about how big is too big, one of the drawbacks is human hearts are radically different in size, and for instance, training, so it messes up your training. Janine, are you doing a lot of cardio right now? I can guarantee you that Janine's heart in total size is smaller than Quincy's. Quincy's probably almost 2 times the mass of Janine. What do you expect, right? The heart's supposed to pump to its muscle mass. And so among things that determine heart size, body size is the biggest. This little tiny sex difference refers to the fact that, remember, males for the same exact weight will have slightly more lean mass, and the women will have slightly more fat mass. And those are both survival characteristics of the species. One is not better than the other. But so you put these two together, and it's mostly like humans tend to have 10 mils of heart volume for every kilogram of lean. That's your best descriptor for the time. It works for children. You can watch a child grow, and its heart will never deviate from that. Yes?
Are the increases in heart mass like linear in time with the increases in lean mass? So like for example,
Yeah.
if somebody puts on lean mass over
Yeah, I can
it would
show you
catch
that
up.
graph, yes. Yeah, during the adolescent growth spurt, yes. Then you'll see that there's type of sport. Interesting. And so we're going to start to look at some of these. So athletic activity is supposed to make hearts bigger. These are elite athletes, almost 1,000 of them. Oh, more than 1,000 of them. Notice that the male distribution and the female distribution overlap greatly. But on general, the females are slightly smaller animals than the males by about 15%. This is what for those of you who had 101, this is a review. Right? So, an interesting thing, I'm going to do a real quick thing with this. If we try to use, this is your cavity dimension, if we try to say a certain cavity is too big, we're trying to use numbers rather than that list of 4 processes, watch what happens. You say, okay, if a male, if you're higher than if you're the top 1% of males, let's use this criterion. Well, notice you're probably underdiagnosing in females because in the female distribution, these 3 gals and probably those 2 have larger hearts by 3 standard deviations than their group. And you're probably underdiagnosing the males. So whenever you put just let's say a heart this big is wrong, it fails. Right? And one of the great problems in basketball, how big do you think Shaquille O'Neal's heart is supposed to be? It's supposed to be huge, right? But it doesn't mean he has a cardiomyopathy. You have to do something more subtle to understand it. So, if you look at electrocardiographic function, you kind of run into the same thing where the variation, see here where this says mildly abnormal, you probably should label this mildly on average. Would be a better way to say it. Because remember, a bradycardia, a heart rate of 53, is quote unquote abnormal. It's outside the range that we set in the 1940s using detrained medical students. But as many of you know, a heart rate of 53 is actually good. It's just on average. So notice that if you, these are different sports. Cycling, you'll see that the highest incidence of cardiovascular, of EKG changes are in highly aerobic sports. So cross-country skiing, cycling, tennis. Soccer is really a sprinting sport. You start to see a drop-off as you go towards the heavy lifting or non-aerobic strength against shooting. So notice that well over half, 50%, in these high aerobic groups have EKG changes, right? And with most of them being routine EKG changes. So the EKG, unless you know exactly what to look for, is also not terribly helpful in determining abnormality. And this follow-up slide gives you now have enough knowledge to begin to understand this. If someone is in a high state of training and they don't show at least 2 of these, I'm a little worried. Because the normal training induces a sinus bradycardia, a long PR interval. There's probably 3 people in this class that have an incomplete right bundle branch block. Because I looked at your EKGs. The early repolarization, I'll bet a third of the class has those ST elevations in the precordial leads with the U wave. Right? On the other hand, there are some things that should never happen. For instance, you can get left atrial. I had left atrial enlargement when I was playing soccer 2 hours a day. Zach Vera, a cardiologist, checked me out when I was a med student, and he listened to my heart. I also had some abnormal heart sounds. He goes, "You're really training hard, right? Because this is intermediate." Now, if an athlete has a left bundle branch block, I don't care how good a shape you're in. That's wrong. Tissue has died. So these different EKG findings are sort of on a sliding scale, and I think we have pathologized these by setting up our standards on a sedentary population. When we set up electrocardiography in the 30s and 40s, we use sedentary people. It's like, well, no, these are variants of normal that indicate they have to be called supernormal. Supernormal, abnormal. That all making sense? But notice how there is no cheap and easy way to say this heart is too big and sick and this heart is too big. You can't use an absolute size. You can use some EKG findings or some declarative. You can, and that's why we do EKGs in many cases. But there's no just, this is the one rubric that always identifies cardiomyopathy. So let's talk about that disease. So this is a genetically inherited disease. And it's defined as when the heart hypertrophies without circulatory or valvular abnormalities. So I like to say it grows bigger for no apparent reason. You don't have a mitral valve regurgitation that's causing you to pump twice. You don't have hypertension untreated that's inducing, I'll show you the difference in a moment, between hypertensive cardiomyopathy and this. So this is just spontaneous growth. And we'll see about why that would happen in a moment. Its genetic prevalence is actually quite high. 1 in 500 of us will have a defect that could lead to the phenotype of cardiomyopathy. The actual incidence of the phenotype is much, much lower, and the actual incidence of sudden death is around 1 in a million. So we have to distinguish a little bit that carrying the gene alone, it depends. We'll talk about this. Everyone tracking? It is genetically predisposed and it's inherited in a dominant, autosomal, not sex-linked, dominant pattern. So this is one of the problems. If you have it in your family line, a bunch of people are going to get it. Achondroplastic dwarfs, you know, the short people, that propagates as an autosomal dominant. So if achondroplastic dwarfs are little people often marry little people. So if the 2 little people have children, 3 of them will be achondroplastic dwarfs, and 1 of them will be a normal stature person who gets the double recessive. All right. So I gave you a little reading. It's from 2014 or 16. Anyway, it's a recent reading. It's a really nice summary of, and I need to tell you that, so here's a bunch of athletes. They've lost a teammate to sudden death during athletic activity. Absolute tragedy. In many ways, I feel guilty presenting this as your first disease because in many ways, you know the term perfect storm from a book of, Younger wrote a book about when every calamity conspires together to totally screw things up. In many ways, as a disease, cardiomyopathy is that because we cannot effectively screen for it. Even though we know it's a genetic disease, you can't genetically screen for it. So to some extent, you'll say, come on, it's 2018, can't we stop this from happening? And to be quite honest, probably not. And learning why that is important in developing your modesty. If many of you think you're going to be in clinical sciences, we need to be modest about what we can do and what we can't do. And unfortunately, this is not preventable. So that's what a dominant allele does. 1, 2, 3, all granddad, daughter of him, daughter of her, all have cardiomyopathy. It's already killed so many people in their family. So once someone dies of it, and on autopsy you find it, boy, you go, there's a good example of where you can be effective. You go and do just like the mom who had inherited hypercholesteremia, you immediately screen the younger family members. So everyone in this family got an echo the first time one person died. That works. Because the incidence of the gene is going to be really high. Let's look at what a cardiomyopathy looks like. So here's a normal heart. Here's normal single cell myocytes all lined up in the way they should be. Here's a normal heart. And if you guys done hearts yet in anatomy? Cool. So here's the left atrium. These are the strings of the mitral valve. The blood kind of flows in. It drops in like this. Then the septum mostly stabilizes, and it's the lateral wall that pulls in from the bottom, like that, to shove this bolus of blood through the canal, right there, and into the aorta. True that? So it comes in. Yes, ma'am.
Once you screen and you, like, let's say the last family that you have the disease, what can you do
well, you'll see that that's another problem. So remember, in this disease, every step is bad. There's no good screening, there's no good diagnosis, there's no good treatment. So I feel a little guilty this being our first disease. You'll be relieved when we get to things. No wonder I'm so happy to take my high blood pressure medicine. It's easy to measure blood pressure, and there's great meds, and then you measure again and they're normal. Hooray! This disease lacks all of that. So good for you for being good-hearted and wanting it to be better than it is. So here's case cardiomyopathy. So first, oh, by the way, remember I did that rule. So there's a wall thickness. 1 wall thickness, 2 wall thickness, 3. Yay, that's a normal ratio. Half a wall thickness. The papillary muscles are grown into big Vienna sausages. There's no, how on earth, the aortic valve is right there. Oh, we have a long path. I was about to say, how on earth can we get the blood to go out there? And the obvious answer, we didn't. Because there's their heart cut out, they must not have lived. Start to see the problem? Also notice that, in particular, we're talking about symmetric in the wall thickness. Notice, for instance, that down here is not nearly as the thickest place is exactly in this canal called the left ventricular outflow tract. So if you wanted to put an obstruction to mess up cardiac output, you couldn't do better than what cardiomyopathy is. Also notice, and we won't get to it today, but this is sort of just setting the table of this nasty disease. Notice what it's doing to the cells. So instead of being, all in a nice line, muscle is sort of like what we call it, pull on a rope, tug of war. In an organized cell, there's one sort of pulled in a direction. Notice these are starting to get disorganized. This is coming in and out of the board. This is probably likely moving. These are still linear, and there's way too much connective tissue, purple, between them. This is not going to pull well, and I promise the connective tissue is going to make it stiff, so its filling properties are going to get messed up too. Horrible disease. So, what causes this? What is going wrong genetically? Most genetic diseases have their basis in the gene expresses a protein, right? So it turns out, and I do not think we yet know why, that whenever you take a primary sarcomeric protein, one of the proteins that kind of organizes the sarcomere, for instance, actin, or titin, or component, if you have a deranged, I think a good example would be myosin heavy chain. I think I told you guys that the complex myosin chain, if you just put 300 of them in a beaker and put it on an agitator and go to lunch, when you come back, it will have self-assembled into the complex myosin molecule. So clearly there's some coding of van der Waals forces along the surface that are causing them to self-assemble. Kind of like the world's best Lego set. You just spray it out overnight and come back and it's built something. Well, if these don't have those layouts, the entire the entire sarcomere chain goes bigger and bigger and bigger. It's almost like I can't assemble this properly, I'm just going to keep working there. So we don't understand why that is, how a damaged sarcomeric protein tells the cell just keep going, but that's what happens. Now notice, if you look at this percent of hypertrophic cardiomyopathies, that myosin heavy chain, perhaps because of its central position in the sarcomere, is very good at creating this disease. And myosin binding protein C is follow up. So you're somewhere around 50 to 60% of all of these diseases are comprised by 2 proteins. Mutations in 2 proteins. So immediately, if you're a good scientist doctor, you think, cool, genetic screening, and we could get 62, isn't that how you think? Prepare to be disappointed. It's the world's worst disease. Yes?
We have problems with the myosin. If the proteins are not performing here in cardiac assets, would they also have problems with synthesis?
That's a really good question. I believe that there are different isosomes. Well, that would be an enzyme. There are different isoforms, different alleles of the same. So usually not. Your skeletal, you couldn't diagnose. That's a really good question. You're thinking, could I diagnose it with skeletal muscle function? And no, not to my knowledge. Great questions. So, we're thinking, let's develop 2 different genetic tests, and we've got this. So you'll see there's 2 problems. The first is, it's not always the same mutation. And the second is, just because you have the genotype doesn't mean you're going to have the phenotype. I don't know how much genetics you guys have, but does anyone ever use the word penetrance with you, where someone has inherited, you could, again, let's use little people. You ever see someone who's like 5'6", but they have the short, stumpy forearms and the somewhat large domed head, and the little bit of bow-leggedness, and you look at them and you think, that person's a dwarf, but boy, they're big. Have you ever seen that with a person? And then there's other people who are utterly tiny. So that's penetrance. That's where they both have the same genotype, but it's affecting the phenotype differently. So first off, look at through time. The difference in expression, if your defect is in the myosin heavy chain, you tend to get the disease early. So this would be the person, the Elk Grove basketball player who dropped dead at age 18. He probably had myosin heavy chain training. Notice if you have myosin binding protein C, it's almost a linear expression, a conversion from the genotype to the phenotype goes up by like 10% every decade. So you're actually pretty unlikely to have a problem until you're in your 50s and 60s. See that? So, and especially think about if you do your genetic screening here, you scare the hell out of 90%, you scare the hell out of a lot of people who have the genotype, and they don't have any of the disease. See what I mean?
Does that mean that their heart increases in size as they age?
Yeah, it grows. Yes. Even worse, let me make it even worse. If you make a genetic test for, say, a valine substituted for a glycine at 46 point, you can actually have different mutations. So for instance, look at the difference in problem in survival. This is a survival curve showing how many people are alive 2 decades, 4 decades, 6 decades into life. So notice, if you have myosin binding protein C disease, which we said does not come on very strong, and you're lucky enough to have an insertion on, it's hard for me to see that, it's like glycine, can't quite see what the substitution is, but all the way out to age 80, less than 5% of the people ever have the disease. On the other hand, if you were fortunate enough to have glutamine substituted for glycine, the 451 position, well, man, you get to age 40 and those people start dropping like flies. Exact same protein, 2 different sites. The same is true, this is myosin heavy chain with a valine substituted for methionine at the 606. That's a pretty benign form of the disease. On the other hand, if your arginine was substituted for glycine at 403, you're pretty well screwed. Excuse my language. So this is a quote from a major clinical researcher in the field. She says, every patient's different. Just because they have the genotype doesn't mean they're ever going to have the disease. And then all these questions that you guys have brought up about how would you diagnose it and how would you treat it, they're all just as bad as what I've just done. It's okay, I promise. There are diseases that are much more easy to deal with. So to finish, just a couple of pictures. So mostly this introduction is about getting an image. So we already talked about normal athletic hypertrophy is eccentric hypertrophy. The hole grows and the wall thickens in perfect proportion so that you wind up, not a very good artist, I didn't know, but you should have 2 to 3 wall thicknesses in the hole. It should be preserved, that relation. And it should be symmetrical all the way around. In pathological hypertrophy, we have what is concentric hypertrophy. Concentro, vamos concentro, los dos. And so the muscles thicken far, far more than they add sarcomeres serially. And so they grow into their own, so which messes up the ventricular filament again. Does that make sense? The end stage of all pathology in the ventricle is to wind up with a dilated heart. And we'll talk more about that when we get to congestive failure. The basic answer is if cells start to die because of the problem, Well then, what happened to the workload on the other cells? It went up, and so they start to die, and then what happens to the cells that are left? So you slowly get an insufficient cell population to cope with the venous return in the heart cells. So, some graphic pictures. Again, this was Dr. Harris's lecture, so it has way too many slides, so I'm trying to cut it down. So again, here's a normal heart. This is cut in the horizontal section. Papillary muscles, evenly distributed wall. Measured there, you got 1, you got 2, and you got about a half more of ventricular cavity measured as wall thicknesses. Here's untreated hypertension. This would be Dr. Shafrath if he went off his meds for a year, right? And you see that this has grown into the hole. You now barely have, the hole is barely 1 wall thickness. This is a concentric hypertrophy due to hypertension. I have to say the good news about hypertension, it's one of the common diseases we'll study. Guess what happens if Dr. Shafrath goes back on his heart meds? This will shrink back to normal, unlike a cardiomyopathy. So cardiomyopathy, this will reverse, just like training effect will. Cardiomyopathy won't reverse. There you go. Hypertrophic obstructive cardiomyopathy. See the problem? The heart is literally muscle bound. That little slip is what you're trying to make your stroke volume. And then ultimately over time, even if you had this and didn't treat it, you would start to lose cells. And so over time, all forms of ventricular pathology head towards a dilated cardiomyopathy. You can get this from overingestion of alcohol. You'll see alcoholics who a pint a day hard liquor at age 60 will start to do this. Doesn't matter the source, doesn't matter who's killing your myocytes, you'll wind up like that. It's like congestive heart failure is the only class of heart disease that's actually still rising because we're getting really good at treating all the other forms of heart disease so people don't die of them, so they have time to heal. You see, I know I'm going fast, but it's actually going to pay back later because a lot of these are just pretty pictures, so I know exactly where I'm going to stop. So we've already seen this growth is not symmetrical in terms of preserving the ratio of wall thickness to cavity thickness. A second kind of asymmetry is that the wall does not grow evenly, and classically it is always the septum growing into the left ventricular outflow tract that is the worst problem. And in fact, if you do in a family identify the disease as dominant, you start doing your echoes, the people who have this variant are the ones at the highest risk for sudden death. They're blocking their own outflow. And so those are the people, Katie, you would try all of our very bad treatments on. But you would at least try because you know they're otherwise going to die. Notice, see the valve leaflets and these papillary muscles? Tell me what's going to happen to this structure as this lateral wall moves in. It's very likely that the anterior leaflet of the mitral valve will get slammed against this like a cork and completely blocked. And so I don't want to overemphasize that because this gets into true medicine. But when you see systolic anterior motion, when you see the structures of the mitral valve being swung up against this lump, those are the ones at high risk for sudden death. That's where if you don't do something, they're probably going to be dead in 2 years, and so you do something. So,
can you explain the difference in expression of the growth of the septum but not the outside?
Yeah, can I not? Because I don't know. It's a great question. And then the last point when we come back, because this is a good place to stop at this slide. So once again, here's a normal heart. There's your outflow tract, left ventricular outflow tract. You can just see the blood going into this canal. Notice how it's not trabeculated. So you're here, trabeculum are great for accelerating the blood out. But once you get to here, you just want a nozzle. It's very smooth. And here's the problem. Now, as I alluded to, this fibrosis is going to create a stiff heart. That normal hearts have a minimum of connective tissue in them. And so all you need to do is relax the cells, and the heart opens widely. And remember, that allows both filling of the ventricular hole, but it also allows blood to be sucked into the capillaries feeding the heart. So that ability to expand rapidly and widely is really important in diastole. This will lack both, and when we come back next time, we'll look at how the fibrosis makes it stiff, so it won't uncoil. The growth has been disproportionate. You've added more ATP consumers in the form of sarcomeres than you have added mitochondria in the cell or capillaries around the cell. And so you've got an absolute setup for insufficient coronary blood flow. And so basically all of the functions of diastole are also messed up. So you can't empty your heart properly, you can't fill your heart properly, you can't get coronary blood flow to the muscles, and even if you did, the muscles are too big and are using too much energy. See what a horrible disease it is? I'm so sorry we started with this one. But there's many causes for hope, and I guess the first one would be that fortunately this is rare enough, it kills less than 1 in a million athletes per year. In terms of bad phenotype that they're intervened with. And we'll talk about why that 1 in a million is probably, we can't get that lower with our current science. We're probably stuck with that for a while. And that's what we'll talk about next time. All right? Very good. So a whole new topic, whole new day. Might even stop raining if we're lucky. They can't have everything. All right. Your TA named Amy is in Banff skiing. Your TA named Saki is home sick. So I am your TA today. I will see you over at lab